Good afternoon, everybody. Still filtering in. Amazing how quickly you showed up here after I got here 5 minutes ago and there were 2 people in the room. So thank you for being so punctual. That's great to see. So today I'm very pleased to introduce Chuliang Song, a new professor at the University of California, Los Angeles. He's been there for about a year now. I knew Chuliang initially when you were a graduate student at MIT, working on some very interesting, really new ideas in theoretical community ecology. So the idea that to really understand the competitive dynamics of a multi-species community, we can't necessarily just be conducting, pairwise invasion analyses as, theory has done for the past 50 to 70 or so odd years. And instead, he and his group at MIT had developed this interesting new approach. It's a geometric approach to understanding sort of structural stability of ecological communities. You can correct me because I'm probably going to butcher the definition of it. But it's really kind of taken off now in the analysis of community ecology. From there, he went to McGill University to work with Andy Gonzalez as a postdoc, where he developed some really interesting tools that are broadly applicable to environmental monitoring, population monitoring under incomplete information, as the title of this suggests. He then moved to Princeton University with Jonathan Levine, working, actually don't know what you were working on with him, but probably something having to do with plant community ecology and modeling, I would assume. And finally, he's here as a TSVP, a member of the TSVP program. I remember when I was writing a letter of endorsement for him, I described you sort of as the Paul Erdos of ecology, given your interest in moving about the world, visiting people, and thinking about really broad problems and trying to solve them. So really great to have you here. And today he's going to be presenting on understanding ecological dynamics with incomplete information. And just another thing, if you want to meet with him, please do so within the next few weeks, because he's only here for a month. So, but he's great to chat with. Thank you. Take it away.
Thanks, Dave, very much for this very kind introduction. It has been a great pleasure to visit Japan. Having been in the US for so long, I really miss all the Asian food. Yeah, just a little bit about my academic trajectory. So I studied my undergrad in pure mathematics. And that journey took a huge turn when I was very fortunate to do 1-year internship at Harvard Medical School, where my advisor was actually a physicist using this statistical mechanics to understand the human microbiome. And that really changed how I view the use of math in understanding biological phenomena. And this really prompted me to pursue a PhD in community ecology. Having come out from a math background, I have to say that I agree more and more with von Neumann, the great mathematician, that if people do not believe that mathematics is simple, it is only because they do not realize how complicated life is. And I guess ecology as a set of life is even more complicated. And something that has constantly struggled me coming from math background in ecology is that usually that collecting data is really tough. It is expensive, and we typically have to deal with limited data. And this has really shaped my research focus on developing those rigorous theories and applicable statistics that we can rigorously extrapolate and predict from those limited data. And as they mentioned, after my PhD, I moved to McGill to study spatial ecology and as well as some potential application to conservation management. And for my postdoc at Princeton, I started working on eco-evolution dynamics. And today I will briefly touch upon all these themes. Okay, now let's take a dive out to nature. I'm still constantly amazed by how gorgeous the natural environment Okinawa is. But the beautiful biodiversity also comes with a curse for us ecologists, as that we have so many species, so much complexity we have to deal with. So to abstract it a bit, the question of interest is typically the dynamics of the whole ecological systems. This is of relevance to fundamental science, such as what are the mechanisms of biodiversity maintenance, as well as applied questions such as how do we conserve these species. However, our data is limited and can be limited in all fronts, such as that on the temporal dimension, how many species are out there, we cannot monitor everything. Are there so many processes at play, such as evolution dynamics as well? So typically, what we have is a knowledge of subsystems. And that begets a big question. How can we infer from the knowledge of subsystems to the dynamics of the whole ecological system? Some ecologists have been arguing that this is a hopeless adventure. Like, you are losing some of the data, which is a truth. And there's no way that we can infer back the dynamics of the whole ecological systems. However, I beg to differ, especially if the question of interest do not require all the data we have, as long as it's usually some coarse-grained dynamical properties we're interested in. As a proof of concept, this YouTube blogger has shredded this photo of this dog into 4 sub-photos. So each sub-photo contains exactly a quarter of data from the original photo. However, if the question of interest is whether this picture is a photo of dog or cat, even if we get 1/4 of data, we can still tell the information. And this is something that we as ecologists, as quantitative ecologists, try to provide these quantitative tools to help empirical ecologists to infer from the knowledge of subsystems to the dynamics of the whole ecological systems. And in this talk, I will show you 3 parts about specific cases of how theoretical ecology can provide helps to deal with these questions. When we go out into nature, we typically see a lot of species co-occurring at the same time. So for example, here, just a hypothetical plant-herbivore interaction network. It's very important to bear in mind that no ecological community is built in a day. They usually have their own observed assembly processes. So for example, here, this plant community may arrive with a certain arrival order. However, the specific arrival orders is usually not deterministic. There can be many processes that can change the specific arrival orders of species. And this begets a big question about how much of what we see about biodiversity patterns would change if we replace the tape of arrival orders. This is not a question that only ecologists are interested in. Evolution biologists have been studying this question for a very long time. For example, this late Harvard professor, Stephen Jay Gould, has asked a question about how much life would be different if we replace the tape of life. But here in the ecological context, what we're interested about is more about the biodiversity patterns. So to abstract it a little bit, what we're interested in is that we want to understand what are the assembly mechanisms driving these ecological communities. And the information we have is usually only one observed history, the specific arrival order of species into this community. However, history is the science of what never happens twice. Then how can we infer from only one observed history to understand what happens otherwise? In very small systems, very small time scales, we can perhaps do this in a lab environment. But if we're interested in these species-rich communities and in those in nature undergoing decades of transformations, it's going to be very, very hard to redo it in the experiment setting. So to show you how theoretical ecology can somehow help with this, we first need to go back to take a little bit of detour about how do we quantify biodiversity patterns. One of the key aspects of biodiversity patterns is really about how robust species coexistence are under environmental perturbations, as the environment is never static. And a key tool ecologists have been using for decades is this tool known as dynamical stability. It basically means that if a system is at equilibrium and we perturb the abundances a little bit, whether it will return to its equilibrium or not. So for example, in this first example, the system would be considered dynamically stable as a small perturbation, after small perturbation, system would go back to its equilibrium state. However, the one on the right is not dynamically stable, as a small perturbation would drive it out of dynamically stable. However, despite the many usefulness of this concept, this approach does make a fundamental assumption in the sense that all parameters are fixed. For example, how strongly species interact with one another, how strongly species can grow by themselves. So all these parameters are fixed. So the environment only perturbs the abundances, but not the intrinsic dynamics. While this may be true in some cases, this is definitely not how ecologists been understanding the effects of environmental perturbations on ecological systems. And this promotes to look at the problem using another perspective called structural stability. So structural stability is a general concept from dynamical systems research. The general definition is just the full range of parameters compatible with some quantitative dynamical behavior. It might be a bit abstract, but we can take a look at this common example of this H2O. So as we all know that H2O would have three quantitative behaviors. It can be ice, it can be water, it can be vapor. And it really depends on the combination of different parameters, such as temperature and pressure. And structural stability tries to quantify what is the range of those parameters would lead to each quantitative dynamical behavior. And translating this into an ecological context, we can envision the parameters as those environmental dependent parameters, such as intrinsic growth rate of species. So intrinsic growth rate means that the maximum growth rate a species can have without interacting with other species. So by the very definition of the intrinsic growth rate, it must have a strong abiotic component, as there's no biotic interactions going on with the definition itself. And the quantitative behavior ecologists are usually most interested in species coexistence, which means that all species in the community can persist over a long term. And then the structural stability we're interested in can be what is the range of those environmental dependent parameters that are compatible with species coexistence in an ecological community. So here I use this as a cartoon illustrations. To make it more concrete, let's just consider a very naive example of two herbivores competing for different plants. So here, each link denotes whether about the diet of different herbivores. So in the first case, we can see there's no overlap in their diet. So there's no competition. So in this case, again, we draw the two axes as the intrinsic growth rate of the two species. So clearly they can coexist as long as each of them have the positive intrinsic growth rate, since there's no competition going on. However, if there is some diet overlap, which means there is some competition going on between these two herbivores, then the coexistence domain leading, the parameters compatible with coexistence will get shrinked. So for example, here we can see that the region leading coexistence gets shrinked because of competition. And we have ways to quantify exactly how much it gets shrinked. And the very beauty of this approach, as Dave had mentioned, is that it's really easily extended to multi-species. The traditional approach that ecologists have been using mostly apply to pairwise cases. It's going to be very difficult to extend to multi-species cases. But here, if we have, for example, a third herbivore, then all we need to do is just to add a new axis, the intrinsic growth rate of the third species. And we can again quantify what is the range of parameters compatible with species coexistence in this system. I'm omitting a lot of mathematical details here, but we have developed an R package called fixed overlap, which allows you to quantify many of these processes easily with the empirical data you have in mind. So, okay, so after this detail, let's go back into the question we had in mind. How do we infer the assembly mechanisms when we can only see data once? So in order to address this question, we use the dataset collected by our fantastic colleagues from this region in Germany, where they have digged through the historical documents that they get the introduction of non-native plants in this region over almost 2,000 years. And based on this, we can try to reconstruct how does the structure of herbivore plant community changes with new introduced plants. For example, at this time step, we have 3 plant species present in this community, and they're associated with 4 herbivore species. And as time gone by, we see new plants established in the community. And this would introduce new links with established herbivores, as well as allow new herbivores to establish in this community. And we can see how this biodiversity patterns changes over this almost 2,000 years. And this is what we observe. The y-axis shows the year, so from year 9 to year 2000. The y-axis shows the measure of the structural stability. A higher value indicates more structurally stable, so it's more robust to sustain environmental perturbations. So as we can see, quite strongly over these 2,000 years, we see a very strong pattern of increasing structural stability. So it means that systems become more and more stable as the community assembles. Of course, we first need to rule out whether this is just because we're having more species. So does this arrival order matter at all? It could just be an artifact because we have more species in this community. So in order to test that, we just simply design a very simple alternative reality where we randomly reshuffle the arrival orders of plant communities. So by doing that, we would keep the number of species fixed at each year. The only thing we're changing, so we're keeping the species richness fixed, but we are changing the arrival orders. So we are eliminating the effect of arrival orders, but keeping the species richness, the effect of species richness fixed. And this is what we found. The y-axis shows the correlation between structural stability and the year. In this observed case, we can see it has very, very strong high correlation, almost close to 0.9. However, when we just randomly reshuffle it, you can clearly see that most of them become a negative correlation. So here I just show you 10 examples from this now example. And if you think about it, it's not that surprising in the sense that if everything kept the same and we just allow more and more species to arrive in the community, no wonder it becomes more and more difficult for them to coexist. So this means that the arrival order must have to do some non-random patterns in how species interact. And to try to understand what is actually the key mechanism, we designed the two alternative realities. Well, we focus on the effect of phylogeny. So to illustrate how does this work, let's imagine here we only have 6 invasive plants, and they belong to 2 families, family X and family Y. And within each family, we design whether it's the first one to arrive in this family as X1, second to arrive in the family X2, etc. This first alternative world, we preserve the family arrival order, but not what within. So for example, here, in this case, we always fix that within family X. So family X must arrive first, fourth, sixth, while family Y must arrive second, third, fifth. However, within family X, it really doesn't matter which one arrives first or later. So we're only keeping the arrival order of the family, but not within the family. And alternatively, we preserve a species arrival order within families. So for example, here, family X is no longer have to arrive like the fourth or sixth of the community, but X1 must arrive before X2, X2 must arrive before X3. And with this, what we found is that, okay, this is what I already showed before, that with a random arrival, we definitely get a negative correlation. And in this first alternative reality, where we preserve the family arrival order, the preserved family orders, what we see is that the observed correlation become higher, but still nowhere close to the observed correlation. However, once we preserve the species order within the family, then we almost perfectly replicate the observed correlation. And this really suggests that the key assembly mechanism seems to be operate within family. In other words, the arrival orders of closely related but not distinctly related plant species seem to determine the observed trend. And upon further investigation, the mechanism seems to be like, because, so for example, the first arriving species of this family tend to become generalist, while the later arriving species within the family tend to become specialist. And this mechanism operates only within family. So for example, the later arriving species, but from a different family, would also tend to be a generalist, but the later arriving species within that family tend to become a specialist. So again, it's not saying that the early arriving species just inherently become the generalist, but it's more likely that the early arriving species takes out the available niches and become generalists, thereby limit the available niches for later arriving species within that family. And we have extended this framework to also understand other ecological patterns. So for example, some of you may be aware that has been a huge competitive exclusions of invasive plant species of the native grass species in the California grasslands. And we have using the structural stability framework to understand what is driving these competitive exclusions in the system. And we are also doing a lot of work on the theoretical fronts, trying to understand to extend, as they mentioned, the two-species assembly framework into multi-species assembly processes, but here with more fine details. So to summarize, when we can only, if all we have is observational data, we cannot perform experiments to replay the tape of life. Then we can use the series to help us to construct those counterfactual analyses to extrapolate those assembly mechanisms. And in the next part, we focus on the limitation about the number of species. So it's been increasingly clear that for biological conservation, we need to monitor how species interact with one another. But as I mentioned in the very beginning of the talk, we can only often monitor some small subnetworks. And this begets a big problem if we want to understand the persistence of the whole ecological network. So for example, here we have 5 pollinator species and 4 plant species. And all we can do is just monitor those subnetworks. So for example, here, just these 2 subnetworks. What the ecologists typically do is that they monitor those interactions in these subnetworks, and they do not have information about the other species. So they just map it into some ecological dynamics and try to see whether the subnetwork species can coexist in isolation without other species. So for example, in this first subnetwork, this 4 species can persist in isolation without interaction with others. However, a big problem with this approach is that it often leads to contradictory results. For example, one subnetwork, they can persist in isolation, but another subnetwork, they can no longer persist in isolation. However, as a whole, we know it's whether they all persist in isolation or whether they persist together or not. So how do we use this information about the persistence of subnetworks in isolation to infer the persistence of the whole network? To address this question, we again use the theoretical framework of structural stability. So as I mentioned, so just as a recap, for example, we have 3 species. So just for simplicity, let's assume that the total community only has 3 species. So we have the 3-dimensional space, and this triangle depicts all the parameters compatible with the coexistence of the 3 species. And to make it even clearly, let's try to map it down to a 2-dimensional space. So for example, here, this triangle space denotes all the parameters that are compatible with the coexistence of the whole species, of the whole community. And every parameter outside will be the parameter leading to non-persistence of the whole network. And we did a systematic analysis from the centroid of this most persistent whole network to non-coexistent parameter combinations. So along these trajectories, we will compute at that. So for example, with that parameter combination, so that makes the whole community persistent. What are the percentages of subnetworks that can coexist in isolation, and what is the percentage of subnetworks that cannot coexist in isolation? And we do it systematically across the trajectory. So to facilitate the comparison, let's just denote the centroid with 0, the most outside as 1, and the boundary as 0.5. And this is what we found. So again, as I mentioned, the x-axis shows some ecological stability of the whole network. From 0 to 0.5, the whole network is persistent. From 0.5 to 1, the whole network is not persistent. And the y-axis is a proportion of subnetworks that can persist in isolation. And let's first see when the whole network is persistent, what happens. So we found that when the whole network is persistent, a large proportion of some networks would also be able to coexist in isolation. This is in direct contrast when the whole network is not persistent. We found that when the whole network is not persistent, the majority of the some networks will not be able to persist in isolation. And we prove this is a general phase transition with random interaction networks. And we can then leverage this drastic statistical difference in their properties to do a Bayesian inferences. If I sample a bunch of some networks and based on whether the some network can persist in isolation or not, I can update my prior belief in whether the whole network is persistent or not. So in some analogy, you can think about this H2O have these three different qualitative states. The whole network being persistent or not persistent at two qualitative states. It's not like the H2O, the structure has changed, but it's really how the organize changes. And this is exactly seems to be this local sub-communities, how they are organized really change the whole network being persistent or not. Okay, but so far this is still just theory. How can this actually be useful to help real biological conservations? And to do that, we have used a very nice data set from our collaborators who have conducted a beautiful experiment in Ireland in Seychelles. So what they did is that they focused on 8 sites. So 4 of the sites are heavily invaded by the non-native shrub species, while in the 4 other restored sites, they manually removed all these invasive shrubs. And then in each of the sites, they monitor how this plant pollinator network changes for consecutively 8 months. So you have both whether the site being restored or not, as well as the temporal information. And this is what we would have found if we can monitor the whole network. So now we have information of the whole network. The x-axis is actually the months. So as I mentioned, we have 8 months. The y-axis is some measure of the persistence. The high up, more persistent, lower, less persistent. This is average among the 4 restored sites, average among the 4 unrestored sites. So it's quite clear that restoration actually works. It makes the whole community more persistent. And not only that, it also recovers some fine detailed natural history operating this system. So for example, we can see the restored sites and unrestored sites start to diverge around month 4. And this is really in the start of the rainy season and the pollination activities becomes increased. And you can see that their difference starts to close, and this is at the end of the flower season. So there's no much differences afterwards. However, it's beautiful. It's a beautiful restoration experiment. However, the cost is that it's so super expensive to do it and very time consuming. So my collaborator, Chris has mentioned to me that it costs like tons of human labor to do this. So we said, like, how about we just use our framework? And let's just, for example, we just monitor 6 plant species. Can we actually find the same patterns? And this is what we found. So of course, we see a more noisy patterns as expected since we're losing a lot of data. But the key patterns still persist. The restoration first works, and we can see it also covers some of these fine-scaled natural history. For example, they start to diverge around month 4, etc. And based on our estimate, we can save at least 50% of the cost in monitoring this community. So far I've talked about if we have a single community and there are many species, we can only monitor some of the species out there. But another common issue we also have is that we have a huge spatial scale and we can only monitor certain patches, but not all of those patches. And we have also extended this framework to infer those so-called meta-population persistence from subsets of patches. And we have also applied to a bunch of mangrove hummingbird data set. We can also find that we can reduce at least 50% of the monitoring cost about the patches. So to summarize, while we cannot monitor everything, like it would be, again, it'd be ideal if we can monitor everything, but if we cannot monitor everything, then it would be useful to establish those links between those from part to the whole. Again, this cannot be deterministic links. We're losing some of the data, But the theory allows us to establish some non-trivial probabilistic links that allow us to infer from those paths to the whole with a Bayesian approach.
Can I ask a question?
Yeah.
Is this conclusion kind of generalizable for many different kind of causal structures of a network, or is it limited to the case where sort of a fractal-like structure exists in this whole network?
Yeah, it's a very good question. So for mathematical proof, what we did is for the random network. So it's like a adult network. We can rigorously prove that the phase transition happens. When we add more network structures to this random network, the proof becomes much more complicated. But we still found that it seems to be the pattern seems to be holding from simulations. But again, we don't know how general it is to beyond that rigorously. And again, we're also adding the dynamical we're adding upon this, it's local vector dynamics. We're also not sure whether if it generalizes to more complicated dynamics. So these are still open questions in that. Yeah, thanks again for the question. So let's move to the last part about the talk. It's about some of the cases one cannot observe all the processes undergoing. So for example, to motivate it, one of the very few universal laws in ecology is that ecological dynamics are never static. It never evolves ecologists. So for example, here is a very classical example of the snowshell hare and Canadian lynx. So as you can see that ecology has been monitoring since like the from 1820-ish, and we can see the persistent fluctuations between the hare and lynx across centuries of dynamics. And from the very beginning of ecology tried to infer patterns from those ecological time series. And this is exactly the motivation of the local dynamics in the first place. So to show you briefly how do people do it, so we typically put some model on trial. For example, we have a local dynamics between this prey and predator. And the first screw up domain focus on those qualitative behaviors. So for example, this is how Volterra actually showed that in this Nature paper published almost exactly a century ago, where he showed that you would have these cyclic behaviors between the prey and predators. This seems to match the observed patterns in these hare and lynx patterns. However, of course, more recently, ecology has been interested in using rigorous model fits. So for example, we can fit the parameters from the time series, whether it's in sample or out of sample validation, and we can see how much it actually fits the results. However, those results, while broadly useful, well, they're very useful, but they do pose a very complicated problem. To illustrate, let's just focus on another real predator-prey experiments from a recent Nature paper where they have been done one of the most comprehensive predator and prey systems in the macrocosms. Of course, they're not lynx and hare, but here just showing you as an illustration of the prey and predator. So here, for example, we have six different fluctuation patterns between them, but it's no longer the exact body patterns we have predicted. However, in this case, can we rule out the possibility that this system is actually underlying driven by local dynamics? And this has been a persistent dilemma in ecology in the sense that, of course, one possibility is that the whole model is wrong, that we should come up with a new model. But some ecologists have been arguing that perhaps the model is just partially wrong. It might work for the prey, but not work for the predator. Or some ecologists have been arguing that they're actually the correct model, but they're just some unobserved biodiversity factors or abiotic factors at play. So the model is correct, you just have encountered everything. And even in this classic hare lynx example, I can find in the literature all three schools of arguments, even for that textbook examples. And this has been a persistent issue in ecology in the sense that so this professor Stefano Elisina at U Chicago has been arguing that if you look at the physics textbook from 100 years ago and today, the length remains roughly the same because people propose this new new models and replace and kills old ones. But in ecology, the textbook just grows even longer because it's simply no way to kill the old models. And this has been a persistent issue about how do we validate or invalidate a model since these all these possibilities. And in this project, we basically borrowed a very beautiful idea developed by Johan Paulsen from Harvard, where they've been initially applied to genetic data, but we see a lot of potential in applying to ecological time series as well. And this model would address all these problems that have been stated before. So here, just to briefly illustrate how this method works. So first, we need to propose what is a model on trial, but we no longer need to propose the whole model. We just need to propose a model for each species. So for example, this is a model of predator on trial. And we're going to partition into two parts. One part called the growth rate, another part called death rate. So just anything that's positive goes to the growth part, anything negative goes to the death part. So we can always partition it. So all ecological models must have these two parts, because otherwise you would just get extinction or just get exponential growth. And then we also need to observe the data. So we know here is how this prey and predator fluctuate over time. And then they're going to use this model growth rate to infer what is the growth rate from the observed data. So for example, here with the growth rate, which is just proportional to the product of the prey abundance times the predator abundance, all we can just do is just at each time step, just do the product of the prey and predator abundance. And we can see how this inferred growth rate changes over time. I can do it for the inferred death rate as well. It's going to be easy in this case, since it's just going to be proportional to its predator abundance. So we just need to copy the same time series of the predator dynamics. And the crux of the method is to look now at the covariance between the predator abundance and the inferred growth rate. So we just compute this normalized covariance between these two values. And it has been proved mathematically that under quite general conditions, if the time series is stationary, it must satisfy. And even if it's not stationary, but still satisfy some cyclostationary, so very weak stationary result, this pattern must hold. So if the model is a true describer of the observed data, these two covariants must be equal to one another. In contrast, if, for example, if we come up with a different covariant growth rate for the predator, and we can perform the same processes, yeah, we'll get a different covariance value. So and in this case, if the two values do not equal, it has a strong confidence that the model is incorrect. And to wrap up, so essentially that it's a very simple method that you have a dynamical equation, you partition into two parts, the gain rate, the loss rate, and you put into this equation. If they are equal, then give you confidence that the model is likely to be correct. Of course, you can never say with certainty, but it gives you a lot of confidence. But if they're unequal, they give you strong confidence that the result is wrong. And to give you a brief test of why we should expect this to be the case, let's just look at what it means for the covariance of the gain rate abundance to be positive correlated. It means that one species have high abundances, it also must have high growth rates. And in order for this system not to go to infinity, it must also mean that it must also have high loss rate when species abundance is large. And this mathematical relationship is exactly captured by this equation. And this branch of math is actually derived from queuing theory. So what people use actually about modeling like in Starbucks or Disneyland. So I find it quite interesting how this branch of math seems to find its application in ecology. And again, another very nice benefit of this equation is that it can be often non-parametric because both covariance and mean are linear operators. So in many cases, you can just cancel these observed parameters. So the test in the end is going to be a non-parameter test. So it's just with the data and with a proposed model, we can fit. We don't need to do any fit process, and we know whether the model is likely to be correct or wrong. And why this method can address this problem I have mentioned before. So first, let's look at whether it can tell us whether the model is entirely wrong or it's partially wrong. It can do that because now the test is for per species itself. So we no longer need to test the whole model in the same time. We can just do it separately for each species. And it's method proven that if there's unobserved value factors, so for example, there's other prey eating this, if there's other predator eating this prey, but they do not directly enter the predator dynamics, it won't affect the dynamics at all, but it will affect the validation process that I've been showing you earlier. And it also works somehow with changing antibiotic factors, not always, but with some weak assumption, with some mediate strong assumptions, you can also cancel this changing antibiotic factors as well. And to put this method to test, we first compiled 18 data sets where there's little evidence for rapid evolution. So basically, there's just a pure consumer resource dynamics going on in these communities. The x-axis is going to be covariance between death rate and abundance. The y-axis is going to be covariance between growth rate and abundance. And on the left, we focus on the predator dynamics. On the right, we focus on prey dynamics. This one line shows the lockup terror dynamics. So as I mentioned, if the lockup terror dynamics is correct, then the data should fall on this one line. If it's not falling on one line, it means that lockup terror dynamics is very likely to be wrong. And this is how these 18 data sets evolved in this community. So each dot represents a different data set. And the trajectory basically just shows that how these two values changes as the times go by. As we can see, despite these 18 data sets all come from different taxa, different environments, from different ages. Actually, one of the data sets was actually collected over 100 years ago. But we're still quite surprised to see that they all fall very closely on this one line, giving us quite strong evidence that lockup terror dynamics seem to be describing the predator dynamics pretty well. Similarly, work on the prey dynamics. Again, we have this 18 prey time series, and this is what we found. They all fall, again, very closely on this one line. Again, giving us very strong confidence that lockup terror dynamics seem to be a good describer of predation or consumption dynamics. Which again, I have to say that in the beginning, I was really surprised by this result, because when the physicists who first developed this method, they applied this method to genetic transcription data, they basically invalidated all around the 10 dominant theoretical framework to describe this. So it's really a very difficult criteria to pass. So I have to say I was very surprised to see that this very simple lockup terror dynamics seems to be describing the consumer resource dynamics very well. But then let's see what happens when there's evolutionary processes going on. So to do that, we collected 13 data sets with strong evidence for rapid evolution. And again, left, predator dynamics, right, prey dynamics. And again, we have this one line. Let's see now what happens with predator dynamics. So we can clearly observe a very different behavior than before. Seems to diverge very strongly from this one line, giving us a lot of confidence that now with rapid evolution going on, we can no longer use lockup terror dynamic to describe it, which is not really surprising. However, what is really surprising is that even though the predator dynamics seem to deviate from this one line, the prey dynamics seem to fall very closely on this one line, indicating that with rapid evolution, we may still use lockup terror dynamic to describe the prey dynamics, which is actually, for those of you interested in ecological modeling, there's been a lot of debate on how do we do eco-evolution modelings. Do we put the evolution on the predator, or do we put on the evolution on the prey? And it's not really about the true evolution in the genetic variation, it's really the phenomenological effects about the evolution. And model has been proposed on either prey or either predator or on both. And they can produce very similar qualitative results. And this has resulted in almost a 2-decade debate about how do we model the eco-evolution dynamics. But here, I think the data has strongly suggested that perhaps we should model the effects of evolution, at least phenomenologically, on the predator dynamics instead of on the prey. Yeah, to summarize, as I lay out in the beginning of the talk, as a theoretical ecologist, I think my job is to provide those quantitative tools to help empirical ecologists to better understand and analyze the data. And here as three case examples, when we are facing the issue with time that we can only observe one out of many possible trajectories, what we can do is that we can use theory to establish those counterfactual analyses for observational studies to understand what is the likely mechanism. The second one is that if there are many species, there are many patches, what can we do? That perhaps we can try to build these probability links to link from part to whole and tell us more about, from our current samplings, about how this whole community likely behave. And third part, for example, with evolution, that in general, with unobserved factors can confound the conclusions. How do we deal with these potential confounders? Well, we should really try to make those model predictions that will be invariant to these unobservables. So we can really make very strong and robust conclusions about the results. So again, like I like to say that, like, again, like it would be great in the end that if empirical ecologists we would have the ability to sample everything, like then we really do not need the quantitative tools. But in the reality that right now that we can only sample not everything, then I think that we still have a job to do. And thank you very much for your attention.
Thanks so much, Chuliang. Time for some questions? Any questions? One back there. Are there microphones? Or?
Ceiling mic.
Oh, great. Ceiling mic.
Hi. Thank you for a great talk. And I was wondering about the last part.
Yeah.
You say that the fast evolution changes are the Lotka, like makes the Lotka buttercup dynamics not applicable to the predator dynamic. And I was just wondering, is this rapid evolution on the predator end or on the prey end? Like which one of them is evolving, and has that make like a difference on how ours result?
Yeah, that is a great question. So let's just take a look back about this model paradigm. So what was the result we got is that for the praise, they see that it still work. But if exactly so, for predator doesn't work. But we do not know exactly what is the cause. So there are two main possibilities. The first one is that it actually has a different functional form, so we have to write a different model. Or the alternative possibility is actually, for example, this delta value or the gamma value is now a changing variable. And why that matters in the sense that if you, 1 second. Yeah. So we use a non-parametric estimate. So here we cancel, for example, unobserved the gamma or the delta. But we can only cancel it out when it's a constant. It's sometimes varying values and we cannot cancel out. So again, we really cannot rule out which is the possibility. My guess is very likely this is actually the possibility where the gamma or delta is evolving, and this is exactly what equilibration models have been a lot of time in doing that. The evolution changes these predation effects. But it also means that for the praise, so the effect that prey gets from the predator seems to be still constant, even after the evolution. The beta seems to be actually still constant.
Another question is about a plot you showed about the subsystems and the persistence of the whole system.
Yeah.
To go back to that.
Yeah, sure.
I just want to make sure I understand it.
Yeah. From the theory? Yeah. Okay.
Does this mean that if you have lower proportion of subsystems that is stable in the whole system, it actually makes the whole system the whole network more stable? Does that mean that you need codependence between the subsystems to make the whole system more stable?
Yes, this is a great question. It's like those subsystems, they are not like isolated one with another. They are like built together. So it's going to be very difficult. For example, if you build a whole persistent systems, but you can try to build a lot of those subsystems that are actually not persistent itself. So so exactly how they are linked together is something we're trying to work on. But at least phenomenologically speaking, this is a pattern we seem to extrapolate. And as the other audience raised a question about it, I mean, I think there's a lot of open questions about this phase transition we don't understand yet.
Thank you.
Thanks, Chuliang. That's really interesting. I've got a question about the kind of next step after this when you applied it to all that data from wherever it was.
Yeah.
Or the Seychelles.
Yeah.
Yeah. So this graph. Yeah. So I really like that the kind of 6 plant species subcommunity was clearly kind of recreating the temporal dynamics there of the kind of persistence. But I was wondering about, it looks like the mean might be a bit higher, and I was wondering if that might be a general pattern across other subsystems. Like, did you look at other, presumably these are lots of different combinations of 6 plants?
Yes, for sure.
Okay.
Yes. Yeah. So this will get if you just randomly sample 6 plant species.
Yeah.
But again, this is a question I always get from Chris, our empiricist, is that when people actually go out and do the subsampling, it's all often not random. So for example, those more abundant species are more likely to get picked up.
Right.
But this is not something we found. Like, we do not seem to find that the more abundant species seem to give you a better predictable values. So it's not like that if you just monitor the more abundant ones, that it's more likely you can get a better prediction value. At least from theoretical perspective, doesn't seem to emerge. Yeah. So it seems right now it really doesn't matter how much. Well, so in this community, I think it has around 20-ish plant species. But again, how many plant species or how many subsystems you want to measure? There's no universal criteria, so it's really some system specific, and also it's a statistical inference, so it also depends on how much confidence you want to have. So, for example, you can still get a quite similar pattern with just 4 plant species, but the average is so large that it's going to be difficult to reach a conclusive result.
Right. Yeah, I was going to ask also why you chose 6 plants.
Yeah.
That kind of answers that. Thanks. Yep. Cool. There can be lots of interesting things to do about looking at the different kind of, if you rather than taking random 6 species community, like deterministically choosing, maybe
so
abundance also.
Yeah. To extend this framework, we are now working with Evan Frick, which I think you know. He has this global plant frugivority network, and we're also trying to use this to infer a lot of patterns, especially about the effect of invasive plants and invasive plants on the persistence of the whole community.
Super interesting. Thanks.
Yeah.
You had a graph, I think it was a bit after this with the inferred growth rate, inferred death rate.
Yeah, sure. Yeah, you mean this here?
Yeah, so on the right, there are 2, you want the 2 correlations to be the same.
We want the normalized covariance to be the same.
Oh, sorry, that's why I wasn't.
Not correlation, yes. Sorry, the covariance.
Yes.
Okay.
Yes.
Just on this one, in something that you discussed at the very end, where the Volter model breaks. Usually, it's said that prey is running for its life and the predator is running for its dinner. So, what will be their evolutionary that might affect the predator model and to make it?
It's actually a great question, and this is something actually OIST. So as I mentioned, again, for this pattern to emerge, there are many possibilities there. So it's going to be difficult to pin down exactly what is happening, at least if we don't do some additional experiment to test it. And especially, for example, in some of the analysis, I think one of those is actually pioneered by a Japanese scientist in the 2003 Nature paper, where actually in their paper they put the genetic variance in the prey dynamics. So many people would assume that actually the prey dynamics seem to be evolving. But again, the log of terra only captures the phenomenological effect. So it's not the exact evolution effect directly. But on average, what is the effect happening there? It's just that phenomenologically speaking, on the evolution effects on the ecological dynamics, it seems to be acting on the predators' behaviors. But exactly what is the pathway, we don't know yet.
I have a couple questions, if you don't mind. So for the lynx hair dynamics, I mean, I know there's been a lot, like you said, there's been a lot of controversy surrounding why some of the cycles are on or off phase, or out of phase in ways that they probably shouldn't be if Lacca Volterra were the were the overarching model. And one of the main things that I think like Krebs showed was that food limitation and sort of the landscape of fear. And, when there are a lot of lynxes, the rabbits were like less inclined to go eat stuff, which led them to develop sort of a tritrophic Lacca Volterra style model. So do you think that incorporating 3 trophic levels into something like this could lead to better understanding of that system?
Yeah, that's a perfect question. And we indeed tested this framework on this lynx hair dataset. And as I mentioned that for that dataset, we have all 3 possibilities explaining. And what we found with the lynx hair dataset is that the hair is described pretty well by the Lacca Volterra dynamics, but not the lynx. And this actually falls with your explanation, where actually the lynx actually have other food resources. It has been created by others. So Lacca Volterra itself doesn't capture the other factors. But for the hair, the main predation sources seem to be the lynx, and that's how they got that.
So then does deviation from that 1-to-1 line indicate some sort of relative magnitude of wrongness of your model?
No, it doesn't tell us which exactly it went wrong. It tells you it is wrong, but not exactly what went wrong.
Well, and I guess my other question was from that 2,000-year data set from I think Germany, was it?
Yeah, from Germany. Yeah.
So what actually explains that crazy shift in structural stability through time? Because 2,000 years ago, it really wasn't that different than it is now. I mean, was it all farm fields that were abandoned, or was succession going on over that time? Any ideas?
You mean how strong this change happened, or
Well, so I mean, 2,000 years seems like it should just be structurally stable. Kind of the null would just be flat, right? If everything were kind of the same. So, what was the fundamental change, in abiotic features or something having to do with population density of humans over those 2,000 years?
So this is something we suspected, but the reviewer said it's too speculative. So as you can see, the key difference happens around year 1500. And this is actually where there are a lot of non-native species introduced because of the great the invasion to the American land, I guess, to American continent. So there are a lot of new non-native species being brought to it. So that actually drives a lot of the key patterns, changing behaviors.
And I guess, is there a link then between this and your results from the persistence of networks? Because I mean, if you make the case that new non-native species are what's driving that stability, then does that imply something?
Yeah, it's actually, so we also ran another test. So in this for those plants, there's actually 2 types the ecologists have been classified. So one of these is self-sustained, one other is ornamental. So if we just separate the 2 parts, you can see that for the ornamental, there's almost no increase of structural stability over the years. It's really the self-sustained species seem to be increasing. And again, as we mentioned, this must be something have to do with the niche, the reduced niche over time. As said, the later running species tend to become a specialist, competing less strongly. And this pattern seems to be really found in the self-sustained species. So I think this must be some selection going on, driving this niche speciation, which cause increase the coexistence potentials.
Oh, cool. And maybe one final question. So, I mean, that 2,000 years does correspond to some fraction of the interglacial, right? Do you think that if you went to that climate,
that's a good question. Yeah,
went north to that new climate,
we have no idea.
That'd be cool.
Yeah, it'd be interesting to see, but it's everything that 2,000 year data is already a miracle. Yeah.
I was also wondering if it might be sampling effort. About like I don't know how many data points there were in that first bit before the transition where it suddenly got. I wondered if that maybe.
Yeah, so before the transition, there are actually not that many non-native species, as you can imagine that. Yeah, but it also can be a data completion issue.
Yeah,
that,
that's what I thought.
So they have been documenting a lot of historical books, as you can see. Like early then, it's really not that many. Yeah. But the pattern seems to be very robust. If we just sample like only 50% of the data we have, we can still see the positive increase. Yeah.
Cool. Maybe time for one more question. Going once, going twice. All right, please join us in thanking Chuliang again. Thank you.
Thank you very much.